Hi, this is Amy Ritter, Scientific Editor with Biopharm International. Beginning on February 25th, 2013, IBC Life Sciences will be hosting their annual Biopharmaceutical Development and Production Week in Huntington Beach, California. This is a conference devoted to the biopharmaceutical development, production, and regulatory issues around biopharmaceutical products. I'm speaking today with Barry Walsh, Conference Director and Project Leader for the IBC Biopharmaceutical Development and Production Week Conference. Thank you for joining me today, Barry.
Welcome, Amy. It's a pleasure.
So as I look at the program, it looks like the conference is organized into two large categories, methods and molecules. Can you talk a little bit about the topics that are encompassed by these two areas?
Sure. The first 2 days of the event will look specifically at the methods that can be applied to specific processes in product quality operations to help companies overcome challenges to improve their bioproduction capabilities. The methods conference track will focus on viral safety, raw materials, process validation and continuous verification, contract manufacturing, tech transfer, and analytical technologies. So attendees of the viral safety track will hear about the new strategies and technologies companies are utilizing to detect and mitigate risk of viruses contaminating their process development and manufacturing operations, and to ensure their viral safety strategies meet regulatory expectations. By attending the raw materials track, attendees will learn how other biopharmaceutical companies and raw material suppliers are developing control strategies to reduce the variability of raw materials coming into their processes in manufacturing operations, as well as learn how biopharmaceutical companies and raw material suppliers can develop more collaborative relationships to improve raw material quality in biomanufacturing. The process validation track will show attendees how to implement the FDA's process validation guidance, including Stage 3, the continued process verification stage, as well as deliver novel statistical methods and QBD approaches for process monitoring and control. During the contract manufacturing and tech transfer track, attendees will learn about proven approaches to contract manufacturing, including CMO selection, building successful partnerships, supply chain management, and how to find practical methods to improve efficiencies in technology transfer during scale-up to ensure process and product consistencies. Attendees of the analytical technologies track will learn how to overcome the major analytical challenges in biopharmaceutical development and prepare to meet the future demands for analytical technologies from companies that are applying novel approaches and innovative analytical technologies to optimize development and understanding, help reduce their costs, and ensure quality and meet regulatory expectations. Then, for the final 3 days, we focused on the molecules and the scientific and technical innovations, development approaches, and manufacturing strategies for the many different types of molecules throughout the product lifecycle. The molecules conference tracks will cover the critical aspects of antibody development and production, including upstream processing, downstream processing, manufacturing strategies, and lifecycle management. In addition, we will cover the development and production of emerging novel molecules, including antibody-drug conjugates, bispecific antibodies, and biosimilars. As MAPs become a more mature class of products and companies achieve or exceed their goals for higher antibody expression levels, we are now facing a paradigm shift that requires innovation in ideas, approaches, and technologies to improve costs, resources, timelines, and quality. These improvements are critical to successfully develop and produce the next wave of antibody-based therapeutics. With that in mind, we have drastically increased the coverage of upstream processing, downstream processing, manufacturing, and lifecycle management to give attendees the in-depth coverage they need to leverage the cutting-edge technical, scientific, and process innovation to improve all aspects of bioproduct development. So attendees of the upstream processing track will learn how to improve productivity, process understanding, product quality, and balance the speed and risk of rapid process development by streamlining the transition from discovery and adopting genomic and other novel technologies. Attendees of the downstream processing track will hear innovative approaches to improve structure, function and understanding, optimize operations, and enable next-generation downstream processes with the evolution of platform concepts, purification technologies, and process analytical technologies. The manufacturing and lifecycle management track will supply attendees with innovative solutions for lifecycle management, regulatory trends, scale-up and transfer, new technology implementation, flexible facility design, and how to implement aspects of the facility of the future in current facilities. By attending the biosimilars track, attendees will learn proven approaches to develop biosimilars for the global market through a critical understanding of market dynamics and regulatory challenges, analytical characterization, and innovative production. And finally, attendees of the biospecific antibodies track will learn the strategies to improve the molecule design, solubility, stability, and half-life, as well as how to reduce toxicity and develop processes for biospecific antibodies.
Thank you. That sounds like the conference is going to cover nearly all aspects of biopharmaceutical production.
Yes, it sure will.
I noticed there's a whole track devoted to antibody-drug conjugates, certainly an exciting new class of therapeutics. What can you tell me about what meeting attendees can expect to learn with regards to this product class?
Sure. By attending the antibody-drug conjugate development and production track, attendees will learn how to apply revolutionary approaches and technologies in early and late-stage development and gain the knowledge to produce next-generation antibody-drug conjugates from industry experts that will share their experiences utilizing novel approaches and emerging tools to help overcome their toughest challenges with linkers, payloads, analytics, characterization, testing, and production to help companies optimize their ADC development programs. Presenters will deliver case studies, unpublished data, and lessons learned that will focus on novel site-specific ADCs, chemoenzymatic generation of site-specifically modified ADCs, concept to clinic in 365 days, design and production of optimized ADCs, next-generation ADCs, characterization and comparability strategies for conjugates, what are the important CQAs for the small molecule, how to design facilities to handle larger commercial batches efficiently, points to consider in the development and control strategies for an ADC, challenges with scale-up safety issues, critical process parameters and tech transfers, implementing an automated preclinical filling line for ADCs at pilot scale, considerations for efficient manufacturing of antibody-drug conjugates, and best practices, regulations, and certificates for handling highly potent compounds appropriately.
Well, thank you for speaking with me about this conference. It sounds exciting and interesting, and I look forward to attending.
It was my pleasure, Amy, and I look forward to seeing you in late February.